just to finalize what I have explained to you, so you have seen that according to these different strategies that can be used, we can select a specific candidate gene, and we can use their sequence to express, for example, the equivalent proteins. But what we observe now is the fact that we don't use only one of these strategies. Very often we combine 2 or 3 of these strategies in order to have more precise number of candidate genes, and by this way it is more efficient, okay, to access to this candidate. So I wanted to illustrate this strategy with one of the part of works we have now doing several years. I have chosen deliberately a part of the pathway that don't lead to vincristine or vinblastine, because I know that some of one of the group is concerned by this question. But I have here chose to illustrate with a part of the pathway that occur in the roots, not in the leaf. Okay, and the alkaloids produced by Catharanthus in the roots are not the same than the metabolites, the alkaloids that are produced in the leaf. But they share the first precursor, tabersonine, because tabersonine is produced in the leaf, and after it is driven across the xylem, phloem, okay, until the roots, and in the roots it can be used as precursor for developing the biosynthesis of several other set of alkaloids that have very nice also biological activity, and in particular locnericin, but also all this acetyl tabersonine and acetyl hydroxytabersonine. So at what moment we needed to have identification of what are the enzymes responsible for the first conversion of tabersonine in locnericin, and also the enzymes that are able to produce these 2 end products of the metabolic pathway. So what we know is that in fact all of these enzymes are quite organized in specific branch of metabolic pathways. So how can we manage the way to discover these 3 types of enzymes? So firstly, what we have developed is some transcriptomic data coming from specific leaf and coming from specific roots. So it could be natural roots, hairy roots, or here it was also adventitious roots. So means that we wanted to combine several new transcriptomic data in order to have the bigger representation of transcripts, not to have just a small snapshot, okay, of the transcriptome in one condition. The strategy we have also after developed is to correlate the expression of the genes in this transcriptomic bank, okay, with one of the genes that have served as a bait. Sorry, it is this one, okay? That was one of the genes that are in the middle of this pathway. And by this way, we have made some correlation with the expressed genes in the transcriptomic data and with different conditions. And in this condition, we find it was quite interesting. We find only 5 candidates. That was the ideal situation, not so many candidates. And in this candidate, it was one that was already characterized in this part of the metabolic pathway. This was a way to prove us that our strategy of co-expression have at least permit to discover, not but to find known genes, okay, in this candidate, which was interesting. And what we have done, we have taken all of the 4 other candidates, and we have developed some study to see where they are expressed. Because we have taken all the transcriptome, including leaf, roots, and so on. So after that, we have also seen where these particular 5 transcripts are really expressed in which organs. So we have tested different parts of the plant. So you can see here that there is flower, stem, leaf, and roots. I have indicated here 2 roots. And what you can see is that effectively this BAIT genes is expressed in roots, but also MAT, which was the second one when we have found this, so it's okay. But here you have 1, 2, 3, or 4 candidates that are also produced specifically in the roots and not in the other part of the plant. So they start to become really good candidates. And after that, what we do specifically in my lab is that in order to test the ability of these genes to function in cells as we want as a good candidate, we transform genetically yeast cells as heterologous system, and we inside the genome of this yeast cell we add the candidate, but also the gene just before the candidate, and in this condition we test the ability of this transformed yeast cells to transform tabersonine in the product, for example, locnericine or other type of enzyme, of metabolite we are waiting for. And this is why after we test several metabolomic study on this specific yeast in order to see if they are able to produce, for example, here it was hydroxytabersonine or different other molecule. And by this way, we have found that only one of these candidates were able to transform tabersonine in the final molecule we want to have. The others were able to produce different other things. And in fact, after that, these genes have been expressed as a protein, and the protein have completely been characterized by enzymology and biochemistry way to declare that now we have already found one of the enzymes that is responsible of this production of the tabersonine derivative 90-acetyltransferase. So this is quite simple in this case because we had only few number of candidates. And this tape, I didn't spoke about this one before, it's a very useful one because yeast cells are very simple system to express plant protein and also to test the plant protein activity in vivo situation, which is more informative than it is if it is in vivo, in vitro situation. And here, after, we have made some complementary works showing that, in fact, this particular enzyme is really expressed only in roots as much as 10-19-hydroxylase, but not like that. That is another acetyltransferase that occur in the leaf to produce the final vincristine and vinblastine. I think people that have presented vincristine biosynthesis pathway have shown this part. It's one of the most famous acetyltransferases that have been already characterized, and we have characterized this new one that is involved in the very last step of the metabolic pathway that occur in roots. But in this same metabolic pathway, it remains also a specific step that was interesting also to decipher completely, it is the transformation of this tabersonin in lochnericin. I saw you at the beginning. The activity that occurs at this moment is an epoxidation. So we are looking for some specific enzyme that have epoxidase activity. So we have done exactly the same workflow. So we have generated new transcriptomic data of our material, but we have included also in this study some other transcriptomic data that come from other type of Apocynaceae plants that are able to produce also lochnericin or some metabolites that are close to lochnericin. The order was to have the maximum of candidates because epoxidase are less represented as a family of enzymes in the genome as it is, for example, for different other type of enzymes. So we have completely opened the transcriptomic raw material of the beginning, okay, in order to have the maximum possibility. And you see here, if we applied exactly the same strategy as we have applied before, we have not only 5 candidates, but a very large amount of candidates. It was 1,526 candidates. So it was impossible to test each candidate to see which one could be the best one. So this is why we have applied a co-expression new analysis by taking not only one bet, but 2. Means this particular enzymes, okay, that occur here, and this one too, that is a related P4050s co-expressed normally with this type of enzymes. We have observed that in fact in several other Apocynaceae, this hydroxylase that is not present at this moment in Catharanthus in roots, okay, could be also interesting to study as a co-expressed, okay, proteins. And by this way, we obtain less number of candidates. Among this one, only 5 are co-expressed with both of 2 hydroxylase. And by this way, these candidates have been already tested in yeast, okay, because we have implemented them directly alone this time in the yeast. When we feed the yeast with the tabersonin, we are only able to produce lochnericin in two clones, in 2 candidates. So we have studied particularly these 2 candidates, and in this case, we have found that both of the enzymes encoded by these 2 candidates are able to make this, okay, this step of the metabolic pathway, so make the epoxidation of tabersonin. So means that in the genomes of Catharanthus roseus, at least there is these 2 genes that could be able to produce the enzymes that will be responsible of this specific step of the pathway. So after these 2 enzymes have been completely characterized at the functional level too, and in fact, both of them could be interested to take in count. What we can do for further experimentation, that have been done after, is that to see if a specific condition of stress, for example, salinity, drought, etc., may stimulate more one of the 2 isoenzymes in order to see if one of these 2 isoenzymes will be more involved in a particular condition, external condition, to see in fact what will be the choice for the plants to stimulate, which of protein can be stimulated, because it seems to have exactly the same enzymatic characteristic, same affinity for the substrate, same capacity, means the fast of the reaction, for example. So the only way that can let us to understand which one could be more the physiological one is to study different conditions in which the plant can be also put. But also we have made a sort of tissue specific analysis to see also if these enzymes are more specific to a specific tissue. But you need to know that not all the tissue are able to produce locnericin-II. So in fact, we need to have an overview of how and why plants have duplicate the activity of this enzyme in particular. So this is always a nice question, and we have not answered to this question for all the genes that have multi-copy, okay, in the genome, and where you can find that all the copy could be functional, in fact. Well, so now we have completely finalized this part of the metabolic pathway, and you can see here that tabersonin that comes from leaf can be in charge directly by this TEX1 or TEX2, okay, depend of which isoenzyme we have, okay. After that, this substrate, locnericin, is taken charge by the hydroxylase, the classical hydroxylase, that after gave some intermediates that are taken charge by the new known acyltransferase, the TAT, to finalize the production of the final alkaloids in the roots. And this is the alkaloid that have the main activity for the plant, obviously, okay, in the roots. So it seems quite, okay, easy to find, but it is also a way to summarize all what I have shown to you at the conceptual levels to see what we can obtain at the end. So it was a very nice work done by a postdoc in my lab. She works about 2 years for that, and she had made a very nice publication for this work. And if you take a look of what we are producing now, there is a lot of new paper, not on this branch of the alkaloid production, but in other plants, pencaminor, for example, not Catharanthus roseus, but also Serpentina revolvia, okay, in which part of the pathway are common, but some specific branch of the pathway are completely species specific. And by this type of strategy, we start to have a very nice overview of the different specific part of the metabolic pathway is in these different Apocynaceae plants to try to find a way how there is a so big diversity of indole, monoterpenoic indole alkaloids in these particular families in particular. And why? Because all of these metabolites have a very strong biological activity, such as anti-cancer activity, but not only, there is a lot of other very useful activity as pharmaceuticals, so it's really interesting, okay, to have an idea of how they are produced. Because in roots, it's like in leaf, they are produced always in a very, very, very small quantities. So if you have a large biomass as leaf in order to pick up some precursor of vincristine and vinblastine, it is not the case with roots. So it means that if some of these roots metabolites may have very important application as pharmaceutical, we need to find a way to produce them by completely an alternative way. And for that, we need to know exactly the metabolic pathway. So this is why we manage this type of works. It is exactly what I said to you. Unfortunately, they are not clustered in the genomes. I said to you that before. And just 2 slides to show, because I don't want to develop something on this area, because the use of metabolomics can be also used alone, okay, in order to improve the biodiversity of this metabolite. And what happens today is that, in fact, we have 2 main methods to conduct metabolic metabolomics with different other methodology, but the two strategy are what we call targeted metabolomics, means that we are looking for what a specific set of metabolites that are already known, okay? So we have this strategy to validate, for example. It is exactly the case in a plant when you knock out a genes, you take all the extract and you do some targeted metabolomics in order to see what are the metabolites that are modified for their production. And by this way, it is quite fast and simple, okay, you know, to characterize, in fact, the effect of whatever modification of gene expression. But sometimes, when you want to discover new pathway, you need to know what are the new potential metabolites that could be produced in a specific organs or in a specific condition in which you have put the plants. So we are able also to develop lots of untargeted metabolites by metabolomics. So by this way, it is a stronger way to discover new molecules and also to hypothesize new branch of metabolic pathway. So it is important also to have these two types of process. It's not conducted with exactly the same methodology. In this case, you may have lots of signals. Most of them are unknown molecules. So after, it's very complex to try to annotate and to identify the corresponding molecule. So what we can do at maximum is what we name network, molecular network, and to assimilate the structure of this unknown molecule with one of molecule that share a part of the common structure that it is known. And by this way, we can at least classify the unknown metabolite more for terpenoids, alkaloids, or other type of metabolites. But after, if we want to go until the identification, we need to shift to another type of methodology, mRNA, for example, RMN. I don't know the translation in English. That could be the final type of method that could permit to identify the structure of the metabolite. But depends of the importance of this metabolite. So here, I can't make a lecture on metabolomics. It is quite complex, and it was not the purpose of this lecture. But take in mind that it is of a very important also in the strategy. Yes, it's a workflow of that. And to finalize, my two last slide is just to open your mind to what happens now. We have now possibility to make this type of study at the level of a single cell. Means that you have seen that for some metabolites, they are specifically produced and accumulate in one type of cell. For example, it was the case for some triterpenoids in trichome of several type of plants. It is also the case for several metabolites in latex, for example. So they are accumulate specifically in sort of cells. And for alkaloids, it's also the case, as they are very toxic. They are mainly produced, the final one, in specific cells, for example, in Catharanthus, is in Latissifer, idioblast, or in Latissifer. So means that when you have developed the strategy to make some transcriptome, and if you have taken at least only the leaf, you have completely deleted the transcript of the good cells with all the other cells that are not able to produce and to accumulate the metabolite you want to study. For example, if vincristine is accumulated in idioblast, when you take a leaf and you prepare all the transcript produced by the leaf, you have the transcript of idioblast, surely, but also of epidermis, parenchyma, vessels, and so on and so on. as idioblasts are only some cells, you can count them in the leaf, you can imagine that how you can dilute the good transcript in this amount of all the transcript of the leaf. So now the strategy that we start to develop is to take only the transcript of the cell in which we know that one of the particular metabolites is produced and accumulated. By this way, the transcriptome of these particular cells will be enriched, okay, with the transcript of the genes that are involved specifically in the metabolic pathway leading to the final metabolites. So that is the new type of technology that now is available, and we have started to develop also this, because by this way it will be a very comfortable possibility to identify really new genes, new original genes, okay, involved in this specific metabolites pathway. One of the examples I saw, it was not in Catharanthus but in another plants, is that by this way they have discovered that the hypothesis of the metabolic pathway they thought before was completely wrong. In fact, there was some other intermediates, and so they have found new genes of new intermediates of the pathway, because single cell technology can be applied for RNA sequencing, okay, but you will see they can also use for metabolomic analysis. So you can make exactly the same type of analysis, but only with samples that come from a specific cells. So it's a very powerful view to identify new metabolic pathway. And if you know that lots of this metabolic pathway occur not only in one cell, but sometimes in 2 or 3 different types of cells, the architecture of the metabolic pathway could be very complex. You can make some single cell technology for at least 1, 2, or 3 types of cell tissue. And by this way, you can confirm some of the metabolic pathway organization, or you can, may have a more precise, in particular, the steps that are between 2 types of cells, to know which are the intermediates that are able to shift from one cell type to another type, another cell type. So here are the summarize of the technology that is now applied to have a transcriptome of a single cell. So the idea, you prepare different sample preparation, and the intermediate is the preparation of protoplasts. Protoplasts are cells where you have to digest all the cell wall. So they are completely separated from the others. And by applying to a mix of protoplasts that come from the leaf, for example, you can apply some screening using, for example, flux cytometry systems, and you can separate all the protoplasts according the origin of the tissue, and so you can have protoplasm of epidermis, of parenchyme, of idioblast, of different type of cell. And by this way, after, you can obtain the nuclei, yes, but also all the transcript, and you can produce some library, and by this way, you can apply exactly the same process as we have seen for normal transcriptome, okay, of a plant or of an organs. And you can do exactly the same with metabolomics. It's very powerful system too. So you can, with micro needles, so you need to have a microscope, okay, and you don't have to take directly the micro needle by hand. It's always, okay, manipulated with micro systems, okay. And by this way, you can take the content of the metabolite of just one cells, okay, that can be linked with transcriptome of this one cells. So it's a very efficient method, because you have just a little drop, and you can inject directly this little drop in a UPLC, in mass spectrometry, MSMS, okay, in order to have directly the metabolite profile of one cell. So you can imagine, okay, that there is now 2 years, from the last 2 years, there is a lot of new publication using this type of methodology that emerge and that comfort some results, but have also shown different type of results. And it will be also a new way to explore how to deciphering some specific parts of metabolomic pathway. I mean, the last one that are the more difficult, okay, to characterize. This is my last slide. Okay, so I know that perhaps I have spoke about lots of things. So don't hesitate to come to see me if you have any question, any comments. I could try to answer to your question. I said to you, I'm not expert in each domain I present today, but I can at least indicate you some paper that could help you to have a well understand of this part of the lecture. But the idea for me is to transmit you what are the modern workflow. And you see this workflow is moving every years, depending on new technology, depending also the precise technology we can adapt. So we need to work very close to people that are producing machine that for us to manipulate all of this. So it's very interesting also. We discuss a lot with customers saying, we would like to have something that permit to do that. And in fact, with this dialogue, we can really improve all this technology. And this could be very useful for the next 2 or 3 years, but I'm sure that new technology will appear, okay, in the future to be more suitable for this type of objective. Thank you for your attention today. I'm sorry if I was not so clear at each point, but don't hesitate to come to see me to discuss if you want. Okay, so now it's your turn to work.